Hi, grade 8. Are you fine? Today we are going to practice about the immunity. In the following experiments, we study the immune response against cock bacillus, which is a bacterium responsible for tuberculosis, an infection in the lungs caused by coccus bacillus, which might lead to death. We have two experiments. In the first experiment, we have two mice, A and B, and in the second, we have C and D. Uh, in case of mouse A, serum of an immunized mouse against coccus bacillus, injection of serum, um, it causes death. Blood cells of an immunized mouse against coccus bacillus are injected to mouse B, it survives, and are injected to C and D, but differently. We take out B lymphocyte, we inject them to a mouse C with the injection of coccus bacillus serum. It causes the death, and the T lymphocytes are injected to the mouse D, and which is injected also by coccus bacillus. It survives. Okay. Pick out from the text the definition of tuberculosis. An infection in the lungs caused by coccus bacillus. Interpret experiment 1. After the injection of coccus bacillus, mouse A, which had been introduced with serum of an immunized mouse against coccus bacillus, died, while mouse B, which had been introduced with the blood cells of an immunized mouse against coccus bacillus, survived. This indicates we must notice the indication, because interpret means analyze and indicate. This indicates that the blood cells protect mouse B from the bacteria, while the serum did not. Describe experiment 2. We separated blood cells of an immunized mouse against coccus bacillus. Then we introduced B lymphocyte into mouse C and T lymphocyte into mouse D. Then we injected both mice with coccus bacillus. Mouse C died while mouse D survived. Referring to experiment 2, show that immunity against coccus bacillus is cell-mediated. T lymphocytes protected mouse D from infection with coccus bacillus, while the B lymphocyte didn't. Therefore, immunity against coccus bacillus is cell-mediated. Another question. Experiment on 2 chicken were done to study the mode of action of and the importance of vaccination and serotherapy. Here we have vaccination of chicken A, vaccine repeatedly after 1 month, it survives. The injection of virulent cholera bacteria in case of serotherapy, chicken B, injection with anti-cholera antibodies, and after 1 hour, it survives. But in case of vaccination, look at the immunity, which develops progressively. In serotherapy, it develops rapidly, but do not last a long time. Copy the below diagram, fill it to compare between vaccination and serotherapy. Write the suitable corresponding letters. A, it provides specific immunity against the microbe. B, provides an urgent, immediate treatment. C, slow developing immunity. D, it develops an immunity that lasts for a long time. E, it provides effect, lasts for short period of time. F, it is a therapeutic, curative method. G, it is the prophylactic preventive measure against microbial infection. And this is the answer. In case of serotherapy, which develops an immunity that lasts for a long time, E, it is protective, effect lasts for a short period of time. And F, it is therapeutic curative method. And for vaccination, it is slow developing immunity. D, develops an immunity that lasts. This is B, for example, this is my bad. It provides an urgent, immediate treatment. This is B, and this is D. And G, it is a prophylactic preventive measure against microbial infection. Draw out from document 2 the importance of repeating a vaccine. It reinforces the immunity, more protection from the microbe. Specify if the chicken bee would survive or die if it was injected with the virulent cholera microbe at week 7. It will die because at week 7, the amount of specific antibodies will be null, vanished. And this is the end of our video. Thank you for watching and goodbye.